Hello viewers, today let us learn about the fate of pyruvate in glycolysis in short. So pyruvate is the end product of glycolytic pathway in aerobic metabolism. Pyruvate is further oxidized into acetyl-CoA. This is a 3-carbon pyruvate molecule in the presence of NAD and coenzyme A forms acetyl-CoA by releasing one molecule of carbon dioxide and NADH. In the absence of oxygen, pyruvate is reduced by NADH into lactic acid. Pyruvate formed in the cytoplasm is transported into mitochondria. Inside the mitochondria, pyruvate is oxidatively decarboxylated into acetyl-CoA. It is an irreversible reaction and the enzyme is pyruvate dehydrogenase. So, this reaction links glycolysis and TCA cycle. This pyruvate dehydrogenase enzyme is a multi-enzyme complex. It has pyruvate dehydrogenase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase. And this enzyme requires the following coenzymes: thiamine pyrophosphate, NAD, FAD, coenzyme ASH and lipoic acid. How does it regulated? Pyruvate dehydrogenase is regulated by allosteric mechanism. Acetyl-CoA and NADH, they are allosteric inhibitors. It is also regulated by phosphorylation and dephosphorylations. So, we know that one molecule of NADH gives you 2.5 ATP. Let us learn about energetics of glycolysis in the separate videos. And we know that pyruvate dehydrogenase occurs along with the coenzyme thiamine pyrophosphate. In thiamine deficiency, pyruvate is accumulated and it is reduced into lactic acid. This leads to lactic acidosis. And now let us learn about the summary of glycolysis. So, glucose by hexokinase and glucokinase in the presence of ATP forms glucose 6-phosphate. Glucose 6-phosphate by isomerase enzyme forms fructose 6-phosphate. Fructose 6-phosphate in the presence of ATP by phosphofructokinase gives you fructose 1,6-bisphosphate. So, this step is irreversible and it is a rate limiting step. Fructose 1,6-bisphosphate by the enzyme aldolase gives you glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Dihydroxyacetone phosphate by isomerase enzyme gives you glyceraldehyde 3-phosphate. So, this step is interconvertible. So, for a molecule of glucose, we generate 2 molecules of glyceraldehyde 3-phosphate. Thus, glyceraldehyde 3-phosphate by dehydrogenase enzyme in the presence of NAD forms 1,3-bisphosphoglycerate. This 1,3-bisphosphoglycerate by kinase in the presence of ADP form 3-phosphoglycerate. So, in this step, it is a reversible step. In this step, we generate a molecule of ATP. So, this is substrate level phosphorylation. 3-phosphoglycerate gives you 2-phosphoglycerate by mutase and 2-phosphoglycerate by enolase form phosphoenol pyruvate. This phosphoenol pyruvate in the presence of ADP by pyruvate kinase in aerobic metabolism it gives you pyruvate and in anaerobic metabolism it gives you lactate in the presence of NADH. So, this pyruvate kinase step is also a substrate level phosphorylation because in this step also we generate a molecule of ATP. That's all about today. Thank you.